All right, good morning, everybody. Today we are going to talk about one of the biggest ideas in biology, and that is translation. Every particle of your body, pretty much, is a translated protein. Now, there's things that aren't translated, like I don't know, some of the nutritional things that you've eaten, some of the fluids and water in your body, but all the proteins that really kind of make up you were translated on molecular level. So your whole body was made, this is kind of wild when you think about it, in these little tiny molecule factories. Molecules are really, really small, but they did enough work to make you. So that's kind of cool. I don't know. I kind of geek out a little bit on stuff like that. So, translation. A lot of people hear translation, they think of like, well, yeah, that's what I do in my foreign language class, okay? So, translation is just taking one thing to another, okay? It's not always about language, okay? So, translation is making a polypeptide. And if you remember back to the beginning of the course, we put together amino acids and called them polypeptides. Okay? It's doing it in a certain order. Okay? Sugars don't need information to assemble like this. Okay? It's just the same carbon ring over and over and over and over again. And if it changes a little bit, that's nice. We have things like starches. We have things like cellulose, where the order is definitely important, but we don't really have to have that much of a structure. It's basically repeating over and over and over again. When you get down to biologically active proteins that have very specific jobs, they have to be built exactly right or they don't work. And so we're going to take that information from the DNA to the mRNA. We're going to send it to ribosomes. You guys, when you've done biology in the past, ribosomes, definition of that little organelle, has always been site of protein synthesis. Well, now you're going to see how that happens, okay? What does the ribosome actually do? A lot of times in textbooks, you'll just see ribosomes as little dots. I want you to start thinking of them more like hamburger buns, okay? Sort of a small bun on the bottom and a big bun on the top, okay? So ribosomes are made out of two sort of parts, the big part and the little part. We're going to get into all the technical names of all that stuff, but you're going to take a sequence of information in mRNA, and just remember, mRNA doesn't turn into an amino acid. All it does is give the directions to do it. Okay? So keep that in mind. So tRNA, which stands for transfer RNA, is going to be the most important thing, key player, in turning that information into an amino acid sequence that folds up into a protein. Now, I just ask you to watch a little video because everything is moving very, very quickly. And without understanding like an animation of it, this can be a little tricky. So I had you watch the video first. So everybody in here just watched that little video of a ribosome sort of going down the strip of mRNA, and then the transfer RNA bringing in the amino acids, and they get put together like a sort of like a string of beads, okay? Remember those little like preschool toys where it would be like a bead, you connect to another bead? There's one up there underneath my clock. That's kind of what we're thinking about here. All right. Let's take a closer look at transfer RNA, tRNA. First of all, which part of this is tRNA? It's this area right here, okay? This right here is your amino acid. The tRNA, oh, I'm sorry, I made that circle too big. This is the tRNA. This is the amino acid. And this down here is the mRNA. All right. So there's three things there. Know which one's which, okay? The tRNA is kind of like a delivery driver that only can bring one product. Okay, think about like your UPS or your mailman. They can bring all kinds of stuff. Think about the TNRA as like a specialized delivery truck. Like cement trucks can only bring cement, okay? There's food trucks that pretty much only bring food, all right? There's, what other kind of trucks? I don't know. Ice cream trucks, only bring ice cream. Only bring a specific amino acid, okay? So, what TNRA does, it has an anti-codon region, okay? So that's going to be the complement to the mRNA, okay? Complementary and anti-parallel. What's that anti-parallel mean? You should probably remember that 3-5 thing, the direction. This is important because, do you guys know what a palindrome is? What is a palindrome? Reads forwards and backwards the same. What are some palindromic words? Wow. Yeah, race car is a long phrase. That's one. Dad and mom. Okay? These have to build in a particular direction, or else we could get some of them flipped around. Okay, so we got to be careful with that. All right, so that's why they have to build in a particular direction. They can't just go in either way. Okay, so they're not truly palindromic, but like if they weren't directional, then ABC would be the same as CBA. All right. Like I said, tRNA is like a truck that brings a specific, okay? It brings a specific amino acid that the codon is asking for, okay? So the mRNA has the code written here for this particular amino acid, okay? THR. THR can't bind with mRNA. So we have to have transfer RNA, bring it in, and connect it to make that big polypeptide. So this is going to connect to the ones that were before it. Okay, and you're going to make these really long strings of amino acids that fold up into proteins. Brian, would you shut that door? All right. There's an enzyme. You don't have to know this one. Amino acetyl tRNA synthase. It's a mouthful. That enzyme attaches amino acids to RNA. So that happens up here. When the thing you do need to know is that when tRNA is carrying its payload that changes its state. Now, biochemically, why is that important? You don't want empty trucks coming to deliver stuff. So when the truck is full, it changes its state. tRNA can only bond when it has that changed state. That's the important thing. Did I say that? That's what I was, whatever I said, I should have said tRNA. There's a lot of, what are they called? Acronyms today. A lot of initials. Keep me straight. I need that. Tells me you're listening. I like it. All right. Thanks for correcting me. tRNA. I got to slow it down when I say that. So why am I talking about this? tRNA has to have its state changed when it has a full payload. What's the payload? A single amino acid. You don't want an empty tRNA bonding with your mRNA. Okay, there's a lot going on here, a lot of initials, a lot to keep straight. All right, I've got some arrows. All right, here's my big hamburger bun. Okay? Here's the bottom of the bun right here. Here's the top of the bun. I want you to think of mRNA as sort of sliding between the bun. Okay? The top of the hamburger bun is like a little factory that tRNA drops into and the polypeptides start growing. If you watch the animation, it's going to give you so much more than my little slide deck here. Okay, so what's happening here? In basic biology, students learn ribosomes make proteins. Well, here's how it happens, okay? First of all, they don't just make them on their own. You throw out mRNA and it'll make whatever you tell it to make. Now, the cool thing is, I could give you the mRNA from a mushroom and your ribosomes will make mushroom proteins. I could do the same thing with whatever, okay? So, any ribosome in biology can make any protein in biology. This is really, really cool. All life on Earth has sort of the same manufacturing equipment, and it all makes this, like, they all read the blueprints the same. Okay, that's kind of the big deal here. Now, it is a little bit different between prokaryotes and eukaryotes. Okay. Prokaryotes have smaller ribosomes, eukaryotes have bigger ribosomes, but they still will make the same stuff, okay? They still read the same code and make the same amino acids. Ribosomes are just a little different. All right, so the two subunits, that's your hamburger bun. Is that too fast? Sorry. All right. Now, if you can remember APE, APE, that'll help you out. A ribosome has three little sections. So that top bun has an A section, a P section, and an E section. The A section is where the amino acid enters. Think of the A as where things go in. Okay? So this is your A right here. The P section holds that tRNA that's loaded. It's kind of like a loading dock, okay? Where the tRNA drops off its cargo. What is its cargo? The amino acid. So look here. What's going to be the fate of this little amino acid right here? This is going to drop in, and then it will transfer onto here, and then the empty tRNA comes out, and what's it going to want to do? It's ready to be reloaded with another amino acid. All right. Lots of things are happening here. Lots of new ideas. You're probably going to have to look at this more than once. All right. This is the kind of question you might get asked. Okay. How does tRNA interact with mRNA? All right. Well, through its anticodon. Okay. If it's GCG, up here it's going to be CGC. Okay? If this is your mRNA, my drawings are getting ugly, but this is your tRNA. All right. Could I go back for a second? Yeah, I can go back for a second. You good now? Okay. I know you take outstanding notes, so I never feel bad about this. I've seen her notes. They are beautiful. All right. Second check question. This is the other kind of question you might get. What actually gives the code for the amino acid? Okay, that's the mRNA. What are the three sites on the large ribosomal subunit, the top of your hamburger bun? That's the APE, the amino acid site, the polypeptide site, and the exit site. It's pretty easy to remember which way they enter because one of them is named exit. Okay, there are the three. Let's talk about these three parts, okay? So, in translation, it's kind of like DNA replication, only instead of making a new strand of DNA or a strand of mRNA, you're making an actual protein, okay? So, here comes our hamburger bun. Here's our ribosome coming together. The top of the bun, called the large subunit, and the bottom of the bun, called the small subunit, make a sandwich out of mRNA. Okay, so what's going to happen here is it's going to bond to it. And remember, charged, why is it charged? Because it's got a payload. It's only charged when it carries an amino acid. It's going to bond to what's called the start codon, which is always AUG. Okay? The code AUG tells ribosomes where to begin and an anticodon for that start codon comes in. So, I think so, yeah. So, proteins always start with this particular amino acid, always abbreviated MET. Methionine. So you're literally building a sandwich. It's not really a sandwich until you have something between the buns. Now, this is really getting down into the nitty-gritty here. But when this first one comes in, it always has to start on the P site. And if you look here, this is lined up. Okay? Why has it got to start on the P site? Well, you can't really, the reason there's a start codon is because you can't really start unless you have something to attach it to. Okay, it's like having one Lego block. Like, I got to put something on it, right? So you got to start with something to attach. Well, the attachment site is right here. So we're going to start by putting it in here. All right, let's take a look about how does this thing get longer. And this actually happens really, really fast. Like, proteins just zip along. So what's getting longer? Okay, does the mRNA get longer? Or does the actual polypeptide amino acid chain get longer? Which one? The amino acid is what's getting longer. So you have your start codon. Your next tRNA delivers your next amino acid, and then you go from the start, and now it's got the first bead, the first amino acid on the polypeptide. And then another, and another, and another, and another. And you get this really long strip of amino acids. Does anybody remember what they do after they're made? They what? Fold. They fold up into these three-dimensional structures. Does anybody remember? Primary, secondary, tertiary, quaternary. That's not what this is about, but that's what's going to start happening. That's what's going to start happening to these chains of amino acids. So we're kind of circling back all the way to the beginning of the course on how they're actually put together. What? Are they added to the P or the A? they go into the A site, and then look, it's like a train going down the tracks, okay? The P receives from the A because the ribosome slides down the mRNA. It's not like a new one comes in every time. Okay? mRNA gets like zipped through the hamburger bun. What's the hamburger bun? The ribosome. Okay? So think of that pooling in there. The ribosome sort of slides down through, and every time it goes forward 3 bases, another TNRA delivers another amino acid. The amino acids disconnect from the TNR and the T. Why can't I say that? tRNA. Okay, they disconnect from the tRNA and they connect to the amino acid that's already built. See how this purple one right here moves here? And remember, that's happening through dehydration synthesis. That's going way back. So, the important thing to know here is each mRNA codon, what's a codon? A set of how many? 3, codes for a very specific amino acid. And these little codon charts are going to be used to determine which amino acid the 3 bases code for. There is some redundancy. Some amino acids have, I think one of them has 4 codes, and they all will make the same amino acid or signal for the same amino acid. Okay, I mentioned this earlier in the lecture, but I want to hit this again. Every single organism uses the same genetic code. So this really supports the idea of all life originating and evolving from some common ancestor. Okay? There's a branch of biology called exobiology. And these are biologists that would wonder what life in space would be like, or life from other worlds. And a lot of them sort of ask, if we found life on another world, would it use the same set of, would it use a triple codon? Would it use the same amino acids? Most people think it would. Okay? One of the ways we're looking for life on other planets is to look for chlorophyll. Okay? Chlorophyll has a very specific spectroscopy feature, okay? Or signature. And so we look at the light from other places in the galaxy, or other places in the galaxy, and try to figure out, could life live there? I don't know. That's a pretty niche branch of biology, because so far there's only one planet with life on it. Of course, that Pathfinder mission, that we'll find out soon here. What do you think? Was there ever life on Mars? I don't know. I have my doubts, but I'm not going to close my mind totally. All right, so elongation happens in multiple steps. First, we have to recognize the appropriate codon. Okay, so the TNRA is going to bond with its anticodon. That's going to enter the A site. I think you were just asking me about this, okay? Next, a peptide bond forms, okay? And the final step is called translocation. Okay, the tRNA in the site moves from A to P, and that makes the A open back up so another tRNA can deliver the next amino acid in the chain. It's called elongation because it's getting longer. Okay? And if you look at step 1 and step 2, we've gone from a single start amino acid to now 2. And next we'll have 3. And we'll just keep adding them over and over and over. Okay, now it's going to get longer out on this end. As always with this material, it's important to know the key players. Okay? So far, those are the amino acids, the mRNA, the ribosome, and the tRNA. Know the structures of know what they look like, know how they're made, and then know what they do. Okay, so this has to have a punctuation mark at the end. We're going to stop this thing eventually. Okay? So termination happens. We have a start code and we have a stop code. Okay? And the stop code tells the ribosome to detach, and it's kind of like I said, like a punctuation mark. There is a start amino acid, but there is no stop amino acid. Okay? There has to be a start one because the next one has to have something to connect to. What does that stop codon do? Okay? It signals for something called a release factor, which hydrolyzes. Okay? And that's going to be a bond that it's going to break that bond. The polypeptide releases. So this is going to be like your ribosome saying, goodbye, protein. Good luck. Fold up. And then all these parts that got this job done disassemble. Your hamburger bun opens back up. That way what? Oh, it's going to go find another strip of mRNA to translate. Now remember, a lot of times students will think of this happening only one time in a cell, like, oh, one little lonely mRNA gets sent out of the nucleus. That's not exactly how it works. It can happen. You know, your nucleus might need a certain protein and it might send out huge numbers of copies. Okay? And ribosomes are going to be making it over and over and over and over and over again until some cellular communication mechanism tells the cell to stop. All right, so instead of doing this with a partner, that would be more of an in-class thing, and all you people at home don't have partners. Let's just go over this real quick, Okay? The steps are initiation, which always starts with that AUG, which brings that met amino acid. Okay? Does it go into the A or the P? The first one always has to go into the P, so the thing that goes into the A has something to build on. Okay? Next is elongation. tRNA keeps bringing the appropriate amino acids that the mRNA codes for, and that strip of mRNA moves through the ribosome, and the polypeptide chain is going to get longer and longer and longer. The key point here, everybody, is that it gets longer in a key sequence, okay? You can't just put any old random amino acids together. If you do them in a certain order, you're going to get useful proteins. If there's a mutation or something there, it's going to mess up that protein's usefulness. We've talked about mutations. Okay? Elongation happens in a couple different steps. The codon has to be recognized. The peptide bond has to be made. And then after that happens, after that bond is made, that tRNA releases its payload, and the ribosome clicks forward and ejects out the empty truck, the empty tRNA. So look up here. This thing started with that start amino acid on it. Once it gets moved along, it gets sent out. What do you think the tRNA is going to do after it gets spent? Is it a one-time use? It's going to go grab another one of its appropriate amino acids. Okay? If you're a vegetarian and you are missing a key amino acid, plants are incomplete proteins. They have certain amino acids in some plants, certain amino acids in others. Okay? Let's say you're a vegetarian, you're like, well, I only like peanut butter. It's going to be missing essential amino acids, and eventually you're not going to be able to fold the right proteins. If you're a vegetarian, you have to eat a pretty good mix of plant proteins. As long as you do that, you'll be fine. Okay? You want to make sure you get complete amino acids? Eat meat. Okay? Milk and eggs as well. Okay? Animal products have all the amino acids. Certain plants don't have them all. All right. Step 3, termination. This is just where it stops. There is no stop amino acid. It just basically tells a bond to break and releases everything, and then the protein will do its folding. All right. I'm going to go ahead and stop it there today.